Hello everybody, my name is Wilmer Roldan. I am junior. Today I go present about mutation in the FLT3 gene, possible biomarker for acute myeloid leukemia cancer. Introduction. First, acute myeloid leukemia, in other words AML, is the malignant clonal disorder characterized by alterations and low production of healthy hematopoietic cells. These alterations exhibit differential of cells and induce proliferation or accumulation of blast. For example, in this picture, you can see the blue stem cell, how produce the red blood cells, platelets, and white blood cells. What happened here? In this part, in myeloid stem cell, is different, change. You can see the color red is the mutation of the cancer. The next, that this year the AML has a new case of 60,530 people and estimated deaths of 23,100 people. AML is frequently a diagnosis in people 65 to 64 years of age. In this graph, you can see in 1992, 2018, the graph in the death rate is down, but the rate of new cases is constant. The two lines is constant. You can see in this part the 5-year relapse survival is 63.7%. Estimate days in 2020, 23 100%. Estimate new cases in 2020 60,530. Previous studies have shown that FLT3 protein has become attractive molecular target for development of new therapies and potential treatment. Mutations in the FLT3 gene have found in patients with AML affecting a 35%. This play important role in how biomarker. In this path, you can see the receptor FLT3. In this is the mutations, the percent of mutation. For example, you can see FLT3 in different percent. Story 3 is years 55, 8% down. Depending the category of years of the person. The next is the graph current is SIIR is one of the large collections of state and regional population by cancer registry. The data represent approximately 30% of United States population. In this graph is that in United States is that 30% this cancer. New case is 21,450 and deaths is 10,920. 5-year overall survival by cancer typing in the United States 2000 and 2016, myeloid leukemia cancer has a survival rate in the half of 50%. In other words, low is the 24%. 5-year survival in United States. You can see the color red is the acute myeloid leukemia is 24%. Okay, my hypothesis is if the FLT3 gene is present in the mutation of the people 15 to 35% with AML, then this may be an early biomarker because this mutation occurs in the people who develop AML. If the results are successful, it can help many people detect AML early because cancer is detected late as it progresses very quickly. Next is the methodology. The methodology for now for the COVID-19 restrictions, the procedures of experiment are going to change. In this, information for surveys and information data that mentor leukemias. The FLT3 will be studied with the mentor base and research aim of the mentor about information of the gene in AML. This I will work in the moment we can pass the restriction of COVID-19 and be able to go to the laboratories to the next steps of the experiment. For the moment, is only taking information of mentor and information of surveys. Study the FLT3 gene to analysis is a stage of mutation. This is work laboratory. And the next is search for antibody as indicator of the FLT3. This work in the laboratory, this is find the shape surface in the antibody to detect FLT3 protein. Run test to see if the AMF detector works. This is I work with a mentor, with two people with AML in Will Cornell Medical. Perform blood tests with a detector to 20 people who have AML. The next is making certain necessary change to the detector if have problem. The next is that people will not be at risk since no chemicals are introduced in the body, only blood is needed, and this is no will be risk. People regardless of age of gender will be used as the test with the conducive with the mentor patients. Collect the data from the experiment with the detector to be able to work more in on that I can relieve to detect the AML in faster. The service will be writing and we will able to the future research and will also be able to have results on mutations in the FLT3. This is the methodology. Participants is I can say 20 people. Is no matter the rank, the race or gender. The participants will corner medical patients. The mentor will have participants 20 patients. Methodology person will ask blood stain performed. Patients will use accessible inks, permissions use. And the risk assignments, I will know risk because it's no introduced chemicals. Protection of privacy, information of the age, gender, or race of the patients will be collect the data. Information consent process, the mentor will share of the informed patients the research is about. The patients will ask permission to access required blood inks for this research. Participation is voluntary, is no required procedures. The next is relate results. These graphs show the expression levels, the AM patients according to FL3 mutation status. Patients with this had significant higher proportion positive blast accompany. You can see the FLT is more than 50% for up in this graph. When a group patients according to genetic characters, the proportion of TD plus AML diagnosis with significantly higher FLT3 possible patients compared with the other. The logistic univariate analysis confirmed the higher TD expression in terms of percentage blast prediction. In this indicate the probability of identifying the mutated patients with TDT expression 69% high in the FLT3. In these graphs you can see it is the high is more 50%. Discussion and conclusion. Our story supports the recently reported involvement of TD3 in the biology mechanisms of primary replication flip page. In primary patient samples, we do not observe a direct correlation between TD possible and FLT mutation status, but TD3 expression levels significantly higher possible in AML. And this supports the possible application of the TD3 enzyme in the generation. The mutation is report colleagues in this line to find trade towards association between higher in this. In conclusion, patients with AML have a higher percent of mutation of the FLT3 gene according to the graph. The 69% identified FLT3 ITD mutations. Importance of application food research. The impact of the FLT3 gene mutation with AML patients is very present who develop drugs against this mutation. That 35% of the people have this gene present, so in future research new drugs can be developed and it can also be a possible biomarker to detect AML quickly and accurately. This is my reference. Thank you.